We are in our fourth of 5 units today, and it is all on bacterial diseases. So it's another grand tour of diseases. So if you liked Unit 3, this one is very similar to that. So if you did well and you like to memorize strange names and diseases, that's kind of what we're going to be doing in this unit. I divided it up. Mostly these are gram positives on this sheet, and then we'll do gram negatives next week. In lab, we are also dividing out gram positive labs with gram negative labs. As far as if you're reading in the textbook, I did pick and choose. A lot of it comes from the chapter 4, which is a sort of tour of diseases and pathogens, and then chapter 15 was the host-microbe interactions and what they do, which I'll give you a little review on that. And then the specific diseases, I have it on the slide. So it's also a digital textbook, so you can also just do the search and it will take you there if you'd like to read. Big thing, as always, study your notes. Use Quizlet, but don't get Quizlet dumb. And you know what I mean by that, just your brain's gonna figure out a way to recognize the card or answer without actually reading what's on it again. So just be mindful of that. Since we haven't discussed bacterial cells for a long time, remember they are prokaryotes, so they don't have a nucleus. Usually there is a circular chromosome in the center. They don't have membrane-bound organelles, and they all have cell walls made up of peptidoglycan. We can divide them into 2 groups, usually by gram-positive and gram-negative. And remember, our gram-positive cells have a very thick outer peptidoglycan cell wall. Gram-negatives are like an open-faced sandwich. Our gram-negatives have a thin layer of peptidoglycan with an outer membrane, and it's that outer leaflet we said was special. I'm going to give you a reminder about endotoxin and lipopolysaccharide. So peptidoglycan, only bacterial cells have that. So sugar chains link together through short amino acids. So that's what a peptide is. For all of these diseases, we're going to discuss pathogenesis. So pathos means disease. So what do these microorganisms have that allow them to cause disease? Epidemiology kind of refers to the who, what, when, why, where, how of disease. But factors affecting prevalence and spread within a community are big ones. And of course, epidemiology is always focused on prevention, if possible. This term prevalence, because I will point out particular groups, this group has a higher prevalence of this disease, and prevalence is just a proportion of a population. I could be talking about Texas, I could be talking about the United States, or I could say worldwide at a particular time point over a specific period. I could say this has a higher prevalence in the spring, or this is, you know, a winter disease kind of thing. But that term prevalence, it's a epidemiology. Let's try that one more time. That word prevalence is an epidemiology term. Incidence is another epidemiology term, and that refers to new cases, like a higher incidence of cholera. Reminding you of our themes, all pathogens have to do 3 things. They have to stick to us. They have to colonize, meaning they need numbers, they need to be able to grow and reproduce and thrive. To do that, they have to avoid destruction by the immune system. To make them a pathogen, they have to do some kind of damage to us. That can either be directly through toxins or enzymes, or indirectly through our immune response. Some microorganisms, it's not the infection, so it's not their numbers living inside the body. They make a toxin that they secrete that will then be ingested, so vehicle-borne, so through food or water. Other microorganisms, they just sit on the surface of our mucosa, so think respiratory tract, gastrointestinal tract, genitourinary tract, and they sit on the surface and they make toxins that affect that organ system. Other microbes go deeper, so they can get underneath skin, or they penetrate through that mucosal lining, and then they make their toxins. And other microorganisms, they don't have a great arsenal of toxins, but it is our immune response to them that does the majority of the damage of the disease. And so I'll be pointing these out to you. The 2 we're going to talk about today are Staphylococcus aureus and Streptococcus pyogenes. That's going to be the lion's share of our day, and then I'm going to hit a couple other streptococci. What you're going to see is one microorganism can cause a whole host or range of diseases. Reason being is that not all strains are the same. They don't all have the same virulence factors. And we've seen this picture on the bottom quite a bit. And in order to be, let's say, like a Staphylococcus aureus, there are a set of conserved genes and phenotypes that's what makes it Staph aureus. However, there are other members of Staphylococcus aureus that will have bonus genes or other non-staph, particularly genes, that give them a different strain. And so we'll talk about different strains that make this toxin cause this. Ones that make this toxin will cause a different disease. So sometimes it's the microorganism that's different. Another huge thing is that we are not all the same. And I think we learned this with our past pandemic. So COVID-19, you have people completely asymptomatic and other people on respirators and dying. So we are not the same genetically. Our immune systems don't function the same. We're different ages, other comorbidities that are going on. So a lot of times it's us that is the difference between what disease is going to be caused. The other thing is location, location, location. Is this in a wound? Is this in the respiratory tract? Is it in the bloodstream? Did it go to the central nervous system? And also, what was somebody's infectious dose? Did they just be exposed to a little bit of the microorganism, or do they have a whole bunch of them actively reproducing? So how many? I thought just for kicks to get a review and to kind of jump back in, because it feels like it's been a long time since we discussed bacteria. So we're going to kind of color our way through that one side of the review sheet. And I also thought, because I'm looking towards the end, so for the final exam, this will be like a great little sheet to pull and go, oh yeah, what do bacteria do? So help you study for your final. So there are two types of toxins. There are exotoxins and endotoxins. Exo means out. Exotoxins are usually proteins. They are either actively secreted or they're released when a cell dies. They can act locally, systemically, and for the most part, most of them are inactivated by heat. Exotoxins can be made by either gram-positive or gram-negative bacteria. And if you look at this picture, what we're looking at is a mucous membrane. And so they have this little sign right here that says lumen. And so you can imagine this digestive tract, respiratory tract, genitourinary tract. We have those two portals of entry, so things can enter through the skin, but the skin is tougher to penetrate, so usually they have to come in through a wound or a micro tear. Our mucous membranes are by far more vulnerable to infection. So I colored mine purple because it's gram-positives day, but like I said, exotoxins are made by both gram-positives and gram-negatives. So they secrete their exotoxins. There's lots of different types of toxins. They can be categorized structurally. We talked about A-B toxins before, and then they can be categorized based on system affected, and we'll talk about those as part of our diagram. But exotoxins are going to act in two ways. Some of them directly damage tissues, so they're directly causing death to that cell, so like a cytotoxin. That's what they're trying to show you in that picture. And the other way is just to interrupt the functioning of that cell. And if they interrupt the function, then they are going to cause a change in physiology. Good example of an idea of that is the Vibrio cholera bacterium. Just sits on the surface in the intestine, and it affects the mucosal cells as epithelial cells, and it changes the way they behave, where they start exporting a whole lot of electrolytes, which leads to profuse watery diarrhea. So they can directly damage the tissue by killing cells, so it's cytotoxin, or they can interrupt the functioning of the cell and disrupting physiology. The exotoxins, they can be released on the surface. They can also be released in tissue fluids, so now we're underneath the mucosa. So toxins can spread. The bacteria don't necessarily need to. Toxin spread is enough to do damage. And then the exotoxin can also be dispersed in the bloodstream. And when we have toxins in the bloodstream, it's called toxemia. Remember, emia always means in the blood, so whatever's in front of it. So like there are some microorganisms, they can be on the surface and their toxins can spread, so in the tissue fluid and then into the bloodstream. An example of this, Corynebacterium diphtheriae causes diphtheria, which is a respiratory illness. So it starts off just respiratory illness. We'll look at it in more depth, but the toxin can then spread, damage the heart. It causes paralysis, or it caused paralysis in children. We vaccinate for diphtheria, so we have very few cases. Still a problem globally. I'm saying the organism doesn't have to go everywhere if their toxin is spreading. That's enough to cause disease at distant organs. Okay, so types of exotoxins. We learned, like I said, we learned about the AV toxin, so the B, the binding part, and then A, the active part. We make toxoid vaccines to the binding part. We also classified them on system that's affected. And we learned that enterotoxins affect the intestine. That's what entero means. And some microorganisms make toxins that cause food poisoning. So emetic toxins that cause vomiting, or toxins that cause cramping, nausea, diarrhea. An emetic toxin is just sort of a subset of an enterotoxin. In the first half of this unit, we got 3 that are pretty big for just producing a toxin that we then ingest. So it's more the toxin than an infection, so food poisoning. So Staphylococcus aureus, the contaminant of food usually comes from the food handler. This is something that is part of people's normal microbiota, some people's. And if they do not have good hygiene, they don't wash their hands before they prep food, then we get toxins in the food. Another one is Bacillus cereus. And it's a cute name because if you just shorten it, it's just be serious. You seriously have some diarrhea and possibly some vomiting. And then the third one is Clostridium perfringens. Clostridium perfringens. Both Bacillus cereus and Clostridium perfringens are gram-positive spore-forming rods. So the reservoir for these two is going to be the environment.
Which two?
Bacillus cereus and Clostridium perfringens are gram-positive spore-forming rods. So we'll talk about how you get those. So Staphylococcus aureus is from humans. So the person that prepared the food for you infected you. And Bacillus cereus and Clostridium perfringens are from the environment and improper food handling. Our next group of toxins are called cytotoxins. And cyto means cell, so these are going to cause cell death. So necrosis, so cell death. And since I was pointing to the lungs, I'm going to use the example of Corynebacterium diphtheriae. It is going to make a very powerful cytotoxin, causes the disease diphtheria. Cytotoxins can act a number of ways. This particular exotoxin acts by shutting down protein synthesis. If you shut down a cell's protein synthesis, that kills the cell. Other ways to be a good cytotoxin is to poke holes, hemolysins, to cut off phospholipid heads, which will result in holes. So those are ways of being a cytotoxin, mechanisms of action of cytotoxins. We also have neurotoxins. Neurotoxins, the ones we're discussing, usually affect motor neurons, or that's what you're going to see clinically. So you can get what's called flaccid paralysis. Flaccid paralysis is when there's no stimulation going to the skeletal muscles. So flaccid, normally people have muscle tone. So flaccid paralysis, if you were to feel their limbs or their muscles, they feel flabby. So that's where flaccid paralysis comes in. Paralysis is paralysis, can't move. Either way, you don't have control. And so the other type from neurotoxins, they'll either induce flaccid paralysis or what's called spastic paralysis. And so this is the other way. This is when the muscles are contracted, but a person cannot relax them. Either way, the person doesn't have control and they can't move, but they will be tense and rigid. So tetany. Samples, bacterium that make neurotoxins that cause flaccid paralysis, Clostridium botulinum, and we'll talk about botulism. We don't have a lot of botulism in the U.S. Most of our cases now are infant botulism, and I'm going to talk about floppy baby. And I'm not doing it in a cheerful way, but if you remember floppy baby, you'll remember. And usually it's from children under the age of 1 ingesting honey. So they get botulism that way. We'll talk about it later. And the opposite in the same genus, so Clostridium, tetany, causes the disease tetanus. We immunize for that. We don't have a whole lot of tetanus. But that causes spastic paralysis. So instead of being floppy, like I said, it causes sustained muscle contraction that a person cannot relax. Danger is always going to be when it goes to the diaphragm, and then you get respiratory failure. All right, how do you feel about exotoxins? Kind of remember? We did kind of talk about it. It seemed like a long time ago, though. All right, so there's exo means out, endo means in. So endotoxins are something that only gram-negatives have. So endo means in. It is something that is intrinsic to being gram-negative. So all gram-negatives. And I went ahead and made my cell pink just because gram-positive stain purple and gram-negative stay pink. You do not have to color yours pink. I'm not even going to see your sheet. So gram-negatives have that thin peptidoglycan cell wall. So we're looking specifically at their wall. So they have a cell membrane, a periplasmic space, and then they have a thin layer of peptidoglycan. And because it was thin, that's why they couldn't hold on to the crystal violet, and that's why they stained pink with safranin. And then outside of that thin peptidoglycan layer, we have a periplastic space, and then we have an outer membrane. And particularly the outer leaflet, because it's a bilayer, the outer leaflet of that membrane is made up of LPS. So LPS, that is the endotoxin. If you forgot, LPS stands for lipopolysaccharide. So we have a lipid A portion, which is actually the part that is the endotoxin. And then we have saccharide. So poly means many, and then sugars, so many sugars. So you have a core polysaccharide, and then sort of variable sugars outside. Yep, all I did was color the arrow. All right, so it's intrinsic to being gram-negative, and what happens is the cells, the LPS can get out into tissue fluid, and how it happens, either the cell dies or they do what's called blebbing. And next week, when I have a great picture from an electron micrograph of Neisseria, which is a gram-negative, and they're blebbing. So blebbing is just when they just pinch off little pieces of their membrane and just, here, have some LPS, just like sprinkling it around is blebbing. So we have LPS, that endotoxin. If they are blebbing, if they're in the tissues, it's in the tissue fluid. Will cause a local inflammatory response. But they can also go into the bloodstream. So they go into tissues, and then they can also go into blood. And if it's in the blood, then it's called endotoxemia. When you have endotoxin circulating in the bloodstream, now we're going to see some systemic effects. If it's just in tissue fluid, we got a little localized infection there. Now it's in the bloodstream, so we're going to see some systemic effects. Fever, so LPS, the endotoxin, can cause fever. And then you're not going to feel good, so just weakness, malaise, feeling just generally kind of crappy. We know our initial response to infection or injury is inflammation. So we're going to get some vasodilation. So capillaries get leaky. And like I said, if it's localized and it's not too much, that's fine. It brings the white blood cells to the area. We get clotting factors out there. We'll get clotting, and it's going to wall off that area of infection. So that's, okay. That's part of our immune response. The problem is if there's a lot of LPS and we start to get really leaky and we have a lot of inflammation, and so then we can have sepsis, right? You hear sepsis, it's always bad, whether it's brought on by bacteria actively dividing in the blood, but you have high amounts of endotoxin, and usually that means there's gram-negatives in the blood, and they're blebbing and releasing their endotoxin. And if that is the case, what you don't want to see is your patient go from being septic, having sepsis, to endotoxic shock. The nursing mnemonic for sepsis is the same letters of sepsis. But the first one, they're going to have a fever, they're going to feel cold, they're going to be shivering, they're going to feel extremely bad, so just painful. They may be sleepy, difficult to rouse. They're going to look pale in color. I'm going to cheat. Oh yeah, I feel like I might die. That part I knew. Sorry. They're going to feel and look awful to you, and then shortness of breath is something else that they may have. So if your patient's looking like that, and you hear your patient, is septic, has sepsis, the danger is it becomes shock, and like I said, it could be from endotoxic shock, toxic shock, doesn't matter. We get that dramatic drop in blood pressure. So imagine all that blood volume leaving, going to the tissues, and then disseminated intravascular clotting, blood flow to organs ceases, right? And then you start to see organ failure. It can happen in the lungs, it can happen in the kidneys. Someone can have blood clotting to their extremities and start to turn black or blue. You don't want to see that. Okay, give you about 3 minutes to finish up your coloring, your picture. Any questions on that? They kind of come back to you?